Good morning, everyone. Welcome to class on Monday morning. There's a slight gender bias in class today. Just a little bit. Yeah, that's really interesting. Well, good morning. I hope everyone had a good weekend, whether that was fun or productive or relaxing, some combination thereof. We got one more week before break. Hang in there. I know it's crunch time, a lot of stuff going on. We got this week, and then you got a whole week off to hopefully have more of that fun / productive / relaxing time. We can do it. On that note, we are going to wrap up our unit on the brain today. And well, let me just jump to the agenda. Here's what things look like. We've got we're going to talk about the white and gray matter of the cerebrum. So we've been talking about the cerebral cortices and different areas of the cerebrum responsible for different sensory and motor functions. We're going to dive into a little more of the brain on an anatomical level today, not get too much detail, but a little bit about areas of white and gray matter. And then we're going to talk just briefly about layers of protection of your brain. And then we're going to wrap things up today with a small group review activity that will provide a good study guide for you for your quiz on Friday. And then I'm just going to wrap up and got some reminders for you. Questions, Zoe?
I know we have the brain review. Is that from visible body?
Brain review questions?
Yeah, it's taking Blackboard. It's doing at 6:30 p.m.
6:30 p.m.?
Yeah.
No, that's the in-class activity. And I meant to do it for 8:30 a.m. So you can ignore the time, and it's not outside work at all. It'll be in class. Yeah, thanks for asking that. I'm sorry it was the wrong time. I was just organizing in Blackboard and put it on there ahead of when we're actually doing it. So yep. No, you're good. Cool. Other questions? Thank you, Zoe, for clarifying that.
Are we having a lab quiz tomorrow?
There's no lab quiz tomorrow. Nope. You have some assignments due. I have a slide at the end with some specific reminders of what's due. So let me just hold off till I get to that slide, but there's no quiz. Yep. Cool. All right. The brain. You might remember this guy. You might have seen this slide a couple times. I've got a study guide to go with the cerebrum if you would like to pull that up. Just as a quick review, we first talked about the brain stem, which is composed of the midbrain, the pons, and the medulla. We then moved and talked about the diencephalon, which is composed of the thalamus, hypothalamus, and epithalamus primarily. We talked about the cerebellum, this guy back here. Lastly, we talked about the cerebrum, which is the bulk of the brain. About 80% of the mass of the brain is the cerebrum. I want to talk a little bit. We've been focusing on the cerebral cortex, which remember is gray matter. So that's composed of the cell bodies and short unmyelinated interneurons primarily. Your cerebrum also has a lot of white matter, and the white matter are fiber tracts, so long groups of neurons together, and they're myelinated. That's why it's white, and they allow different parts of the brain to communicate with each other. And I want to break down a couple of these structures for you today. So you can see this is a cross section. So this is a coronal section, as if a brain is looking at you and got sliced this way. And you can see the lateral ventricle here. You can see the cerebral cortex following those gyri and sulci around the perimeter of the brain. And you can see in the middle here all of this white matter. And it's white. It looks white. And you'll see when we look at actual human brains and sheep brains in the lab, you'll see that it looks white. Now, the white matter, as I mentioned, is myelinated tracts. It's that protein, the myelin, insulating the axons of those neurons that give its white color. One type of tract is called a commissure. Commissures connect the left and right sides of the brain. And specifically, there's a structure called the corpus callosum, which is the largest commissure and which primarily provides communication between the left and the right hemispheres. So these are fibers that are running kind of across the hemispheres of your brain. And specifically, there's a structure here that's just kind of superior or dorsal to the ventricles. That is this white matter that's connecting the left and right hemispheres. So we talked a little bit last week about how the left and right hemispheres have different functions and how often their sensory input and motor output is contralateral, etc. It's important to note that those movements are very coordinated as well, and the communication between those left and right hemispheres happens through neurons, the axons of which travel through these commissures, and especially through this corpus callosum. When we dissect brains, you'll see this corpus callosum really, really clearly. Actually, this image isn't the greatest. I'll show you a nice image of it here coming up. So that's the first. There's three white matter tracks we're going to talk about. The first is commissures, which connect left and right hemispheres of the cerebrum. The second is what's called association fibers. So this is white matter that allows communication between different parts of the same hemisphere. And so, for example, I gave the example here of the areas of the cerebrum responsible for understanding speech and producing speech. We also talked about like the prefrontal motor cortex, or the prefrontal motor cortex and the primary motor cortex, where you plan your motor output and then you execute it. Those areas are communicating with each other. They're on the same hemisphere, so that communication happens via these association fibers. So commissures across hemispheres, association fibers within a hemisphere, and finally projection fibers run vertically to and from the cortex. So this is fibers that are essentially communicating from the cortex down through the brain, primarily through the thalamus, and then to other parts of the brain on down to the spinal cord. And that's all I'm going to say about white matter. Primarily, I want you to know those three kinds of fiber tracks: commissures, association fibers, projection fibers. Questions? All right. Now, in addition to, I'm going to back up for a second here. In addition to the gray matter that is the cerebral cortices, right, on the outside, and we have this white matter I just talked about. Now, there's also some areas of gray matter that are deep in the cerebrum as well. And those areas of gray matter, just like the cerebral cortices, are formed by cell bodies of neurons, as well as these short unmyelinated interneurons. And there's a couple of different areas that I want you to know. The first is a number of nuclei that are referred to as basal nuclei. Specifically, we have the caudate nucleus, the putamen, and the globus pallidus. Are they on this diagram? You'll see these. Okay, so you'll see these in lab tomorrow. Let me think about what I want you to take away from this slide. So this is not the greatest diagram. But here's what I want you to know for lecture. I want you to know that there are these areas of gray matter that are deep in the cerebrum, and I want you to know that they are related to functions of motor control, memory, in some cases, the colostrum. We don't know. That's probably something important. We don't know. I do want you to know the amygdaloid body. That is part of your limbic system. Remember, that's important for processing emotions, especially. We talked about that a little bit last week. I'm actually not really happy with the way I'm presenting this information on this slide, so I'm not going to ask you, at least at the moment, to necessarily know great details about these. Just know that there's areas of gray matter that are referred to as nuclei. Know generally what they're responsible for. In our brain lab tomorrow, we'll get into more detail, but for lecture, let's just do a superficial overview and just be able to recognize the deep gray matter of the cerebrum and know, you don't need to know exactly which ones are which, but just be able to recognize it. So if I gave you a diagram like this, you would know that like this gray matter here is not part of the cerebral cortex, but is that deep gray matter. I cut out some information from the slides and I'm not happy with the way I'm presenting here. So I'm going to just scale it back and not ask you too much detail. Cool. All right, that's all we're going to talk about with the cerebrum. I said it was going to be quick, came through. What I want to move to now is layers of protection of the brain. And there's four primary structures that protect your brain from injury. They are the skull, meninges, cerebrospinal fluid, and the blood-brain barrier. We're going to walk through each of these in a little bit of detail, but in all honesty, not too much detail. What I do want you to remember is that the brain is very, very delicate. And so it has to be very well protected, both from physical injury, but also from chemical insult. And also the brain is super energetically demanding. So we need to protect it from essentially a lack of resources. And specifically, there's two things I'm thinking of that the brain requires to fuel its energetic processes that are super important, that it has to get at all times. And if it's short for even a short amount of time, you can suffer dire consequences. What are those two things it needs? Go ahead, Ed. Oxygen and glucose. Yep, exactly. So the brain primarily relies on glucose as its energy substrate, and it needs both oxygen and glucose to metabolize the glucose, form ATP, and do all its brainy stuff. That's a scientific term, by the way. Cool. All right, let's talk about each of these. First, I want to talk about the meninges. The meninges are these layers of connective tissue that essentially form the barrier in which or this thin layer in which the cerebrospinal fluid sits. And we have three layers of meninges from superficial to deep. We have the dura mater, the arachnoid mater, and the pia mater. And you can see those on the diagram here. We have where's my dura mater? So while this has periosteal and meningeal dura mater, I'm not going to ask you to distinguish that. But I want you to know that as dura mater is essentially just deep to the skull. And actually, I'm not going to, I mentioned here the skull as a primary structure protecting the brain. I'm not going to talk about that too much today because we've covered that pretty in depth already, and I've talked about how it's protective. So we're going to talk about these meninges first. We have the dura mater superficial. Then we have what's called this arachnoid mater. It's called arachnoid mater, comes from the same root word for spider, like arachnid, because it looks kind of cobwebby. It looks like a spider web in here. There's these fibers that cross between the superficial and deep layers. And then we have here, where's my pia mater? Oh, right here. We have the pia mater, which lies here right along and is continuous with the surface of the cerebrum. Notice that the pia mater follows these gyri and sulci of the cerebrum, right? And so it follows these grooves and indentations and bumps along the edge of the cerebrum. So in between this space, in between the pia mater and the dura mater, we have this space, this arachnoid space where we have cerebrospinal fluid. Notice there's blood vessels traveling through here as well. Notice that we have these features called arachnoid granulations, where little bits of this kind of come up and interact with the cerebrospinal fluid in the ventricles there, or in the, excuse me, not the cerebrospinal fluid, that was the wrong thing to say. That interact with the sinuses that are filled with blood. So there's exchange of nutrients and proteins, etc., between the cerebrospinal fluid and your blood. What else did I want to say about this? One other thing, notice that there is in one place in the longitudinal fissure, so that's between the hemispheres of your cerebrum, there's part of this dura mater kind of comes down and goes between those hemispheres. This is called the falx cerebri. I do want you to know that. And that actually attaches, do I have this on my next slide? That attaches to a structure on the ethmoid bone. Oh, I do have that here. And kind of helps anchor the whole brain. So the falx cerebri comes down and anchors to this crista galli of the ethmoid bone. So you might remember that when we talked about the skull. It's that little kind of peak on the top of the ethmoid bone. So this is the same diagram from a different view. This is a mid-sagittal. I don't know if it's exactly, no, it's not, yeah, no, it's mid-sagittal. And you can see here those structures again. What I want you to take away from this is we have this falx cerebri. So in this view, that layer of dura mater that kind of came down between the hemispheres here, if you look at it from the lateral view, you can see it's this big sheet here, and it's anchored to this crystal galli of the ethmoid bone. Essentially, you can think of it like this sac around your brain, and that sac is tied down at one point to your skull to kind of keep it all in place. We also have this structure called the tentorium cerebelli, that translates as the tent over the cerebellum. So this is this layer of tissue between the cerebrum and the cerebellum. And then we have, let's see. No, that's about all I want to cover in that. Questions? Okay, let's talk about a little more about these meninges. The arachnoid mater, as I mentioned, is deep to the dura mater, and it has these special projections that allow cerebrospinal fluid and blood to exchange proteins or other small molecules, et cetera, et cetera. And then I mentioned, actually, I got to hype myself a little bit. The pia mater is what clings tightly and follows those contours of the cerebrum. So I mentioned a couple times the cerebrospinal fluid. I want to talk a little bit more about what it is and what it does. First of all, the cerebrospinal fluid, or CSF, or CFS, excuse me. Oh wait, no, that's wrong. I should fix it. Sorry, that should be CSF, cerebrospinal fluid. Is formed by, gosh, I've shown this slide so many times, I've never noticed that I had these letters reversed. It is formed by these structures called the choroid plexus, which are in the ventricles of the brain. And so I'll just skip ahead. I'll come back to this slide, but I just want to show you where they are. For example, here, in association with those lateral ventricles, you also have one here associated with that fourth ventricle, just dorsal to your brainstem. And here we have these special ependymal cells. You might remember that from when we first started talking about the nervous system. They're one of the 4 types of support cells in the central nervous system. They are a kind of epithelial cell, and they form this layer between the blood vessel and the space where we have the cerebrospinal fluid. And it basically filters the blood in such a way that only the stuff that we want in the cerebrospinal gets through. We produce about ½ a liter of cerebrospinal fluid a day. So picture that, if I have this bottle, that's about this much cerebrospinal fluid is getting produced every day. But at a given moment, we only have about 150 milliliters present. So this includes all the cerebrospinal fluid around our brain, in our ventricles, going down the central canal of the spinal cord. All of that together is only a little over 100 milliliters, which is not much fluid. So these are very, very thin kind of layers of fluid, but they're very important at the same time. Go ahead, Jada. Mm-hmm.
Is that like when you have fluid in the brain, right?
Yep, that's exactly it. Mm-hmm.
It's like more than a liter that's in your blood at the time.
Yeah, great question. So what it is actually, it's an infection of the meninges, right? Meningitis is when those meninges are infected and they get swollen. And so these layers get infected with, I believe it's a bacterial infection, if I'm not mistaken. And because they're infected, they get swollen. You have a buildup of fluids, et cetera, et cetera. But what's super dangerous about that is when you get too much fluid in here, it's not necessarily cerebrospinal fluid, it's fluid associated with the infection. But your brain has nowhere to go. If you put too much fluid in there, you've got the skull on one side, that ain't going anywhere, and then you've got the brain on the other side, which is I say more pliable than the skull, right? And so the danger there is you build up this fluid and you literally squish your brain.
My mom had to get spinal fluid taken out when she had meningitis. Does that, when they get the spinal fluid taken out, does it affect the meninges? Yep,
that's exactly. So basically they have to drain these pockets of fluid so that it doesn't get too much and relieves the pressure basically. Yeah, it's a great question, Jade. I'm glad you said that. Yeah, don't get meningitis, it's bad. And that's exactly why it can be so dangerous. It's not necessarily like a terrible infection. It's just that when infection happens in that place and swelling happens, if you get swelling in other parts of your body, it's not fun, but it's no big deal. You have room for that swelling to take place, but in the brain, not so much. Yeah, exactly. Okay, so we got cerebrospinal fluid. Let's take a look at this. What I want to emphasize, this is a diagram from your textbook. The takeaway message here is I don't need you to memorize the path of the cerebrospinal fluid. What I want you to note here is that it's all continuous, meaning that the cerebrospinal fluid is around the spinal cord It's in the central canal of the spinal cord. It's around the cerebrum. It's around the cerebellum. It's in the two lateral ventricles, the second ventricle, the third ventricle, and the third ventricle and the fourth ventricle. And it's all continuous, meaning that fluid is continuously flowing through all those different compartments. Yeah, I've got a little more detail there, but the takeaway message here is that the cerebral spinal fluid is continuous both around and within the brain. The book has a little more detail about where it's produced and how it circulates, but I don't need to go to that level of detail right here. I do want you to recall that these ependymal cells have these cilia that can wave and they help keep that fluid circulating. So it's important that that fluid is constantly circulating so that it can remove waste and deliver nutrients, etc. And part of the way it does that is by these cilia associated with the ependymal cells. Now, this layer between the cerebral spinal fluid and your blood vessels formed by the ependymal cells is often referred to as the blood-brain barrier. I'm sure you've all heard this one before. This is it's a selective barrier, meaning that certain things are allowed through. So for example, oxygen and glucose, you want to get to your brain. But it prevents certain toxins and larger proteins from entering the brain. And so the capillaries here are relatively impermeable compared to those in other places in the body, meaning that less stuff can get through than in other places of the body. Now, what is a common misconception is that this is like a hard barrier and nothing can get through. That's actually completely untrue. Lots of stuff can get through, especially lipid-soluble molecules. So oxygen, small molecules like oxygen can get through, but also things like alcohol gets through. That's why consuming alcohol affects your brain, right? Nicotine, that's why consuming nicotine, cigarettes or whatever form affects your brain. Anesthetics, which is why they can use those medicines to make you unconscious for surgery and things like that. If those things couldn't get through that barrier, that would be a problem. So certain things get through and certain things don't. And what I want you to take away from this that is a very selective barrier, but it's not an absolute barrier. I'm just backing up for 1 second. Let me make sure I covered what I wanted to cover. Yeah, that's about all I want to say about that. Cool. Questions? All right. What I want to do is as you know, you have a quiz on Friday on the brain. So everything we've covered so far in the brain up to and including the protective layers of the brain. I randomly assign you to groups of 3. And what I'd like you to do is work in your small group and complete this short survey. There's room for each of your names on there. And it is basically your group has a certain part of the brain. So for example, Ran, Juliet, and Gia are working on the cerebrum. Also, Jada, Matt, and Marge are working cerebrum. There's so much to say on the cerebrum that I split this up to 2 groups on the cerebrum. Then we have the cerebellum group, which we're missing. No, Drew's here. Holly, Terrence. So let me make sure we've got people from each group. So you guys are all here. Good. Abby, Kylie, Sophie's missing. You should be a group of 2 though. That'll be fine. Zoe, Jessica, Morgan are here. Travis is missing. So with Veronica and Sarah, you guys will be a group of 2 as well. So go ahead and work in your group. I don't have a timer. I'm not sure how long this is going to take, but it's a chance for you to formulate some review questions. I will consolidate these and then share them with the class on Wednesday, and then we'll essentially build ourselves a class study guide for a quiz on Friday. Yep, so go ahead, move around, do your thing. I can go and check in with you. as you're working. I'm going to pause this recording. All right, I'll bring this back for a second. I'm going to try to think of the best way if I want to wait and share this with you, or maybe I'll just wait and just put these together for you for Wednesday, and then you'll have kind of a study guide for Friday. Overall, it looks like these are really great questions, and the diagrams you've chosen are applicable as well. As you were doing this and putting this together, what questions came up that things you're unsure of, or questions you might have for me? We're good? Cool. All right. Well, let's study that brain. As some reminders, that brain quiz will be Friday. It's closed book. It'll be in class. Keep that in mind. Remember that the closed book quizzes in class have produced lower scores generally than the online open book tests, which is not surprising. But we are closing in the end of the semester. A lot of you are on the cusp of maybe getting the grade you want or a little bit less than the grade you want. So just some extra motivator to make sure you're preparing for that. This week, you'll see in your class program for Wednesday, the lab guide is now available in Visible Body. Our lab tomorrow is the brain lab, which is a lab I really enjoy. We get some sheep brains out. I have some really nice sections of human brain to look at. It's a pretty involved lab, so plan on, there's a lot to do with this lab, and it's available in Visible Body, so read it over before lab tomorrow. It'll be helpful for you to know what's going on before you go in there. So reminder, your quiz on the sensory lab from last week is due before lab tomorrow, and that is in Blackboard. See your lab guide for that, but make sure you get that done. Also, 2 things to bring with you tomorrow in lab that you had assignments that I haven't checked off yet, because I want to sit down with each of you individually. That is your skull drawings from lab 2 weeks ago, as well as the activity sheet on the special senses. You can do that electronically or paper. Either way is fine. If you did it electronically, you don't need to print it out. You can just show me. Either we can pull it up on your laptop, or you can email it to me, or whatever works in class. The reason I'm having you submit these assignments this way is because I like to really sit down with you and walk through all of this, make sure you've got good understanding. I feel like it's a more meaningful way to check your understanding than to just grade it and give it back to you. So when we can, I like to do lab assignments that way. So bring those in. Those are all due tomorrow. I have open hours today, noon to 2 in our lab room. And then don't forget that Danny has his tutorial sessions today and on Thursday. The session on Thursday, I especially want to recommend in advance of your quiz on Friday. That's all I got for today. We're 10 minutes early, which is weird. Usually things take longer than I planned for. So there, you make good use of this 10 minutes today. Go get yourself a cup of coffee. Actually, that's what I'm doing. I'm grabbing a cup of coffee. I'll see you over at Starbucks.